Get share going. A couple of things I do want to. All right, almost everybody's here. All right. I'm going to do a quick look at this karyotyping activity, and then we're going to do look at the Georgia Milestone practice test. All right. Yeah, can I open this? First of all, before we do that, is Talacia in? Yeah, okay. All right, my waiting room now is empty. Okay. Good, good, good. Okay, why won't it open for me here? Oh, I was trying to open the message. Let me go back to that. There's some, how they call it, housekeeping business that we have to do first. If you haven't checked, I have to come out of student view to get it to open. But look at the announcements. It's very important as we're coming up on having to do the, all right, so check your announcement here. Because you have to use your Chromebooks to do the milestone test, it's important that you check to make sure that it's fully functional, that everything is functional. You don't want to have any major damages, cracked screens, keys not working, sound not working, because you must have sound for your ELA test. So any damage to your Chromebook, you need to report that to Miss Card right away. Now, you might have got this message from your other teachers, but I'm obligated to tell you also. So to make sure that you do that. So click on this link and it'll show you how to go through your Chromebook to make sure everything is functional. Because again, you'll have to bring your Chromebook to the site where you're going to test, and they won't have very many extra Chromebooks on hand if yours isn't working. Okay. And then you don't want to have to be there and then have to come back another day for the makeup test because you weren't able to take the test that day. So it's real important that you make sure that your Chromebook is checked. Okay. All right, so that's that announcement. So again, make sure, and folks that just logged in, I did get focus mode fixed, so you can be on camera now. So please do so. And make sure that you check out this announcement. All right, back to the home page. So a couple of you I was pulling folks out while we were doing the benchmark to help you with the karyotyping activity. Whoops, I didn't mean to do that. Okay, let me come out of that. Close, close. All right, so whoa, where'd it go? You click too fast, things happen. All right, so basically that closed. That's why I can't see it any other way. So let me go into the module. All right, so this is the week before spring break. So all we're going to do this week is do the practice exam for the milestone, and we'll be doing that going forward. So you want to make sure that you go back and look at vocabulary for everything that you have done already. And then we're going to look at, after the break, we have not done, I'm going to close this so I can focus. All right, so this was the last one we did. I'm going to close that. And we're going to, after the break, we're going to go do a real quick, fast study of, okay, let me write down these late people first before I let them in. All right, good afternoon, good afternoon, good afternoon. I did fix the focus mode so you can be on camera. So please do be on camera. Um, and then check your message, your announcement page, please. That's what we were talking about. And now we're going to, the last two things that we need to go through are evolution, taxonomy, and ecology. And we're going to kind of go through them real quick. I'm going to touch on the things that are most important before we get to the milestone test. But I need you to be going back and reviewing. So after I go through the karyotype activity, I'm going to take you to a link that we can do the practice test. All right. All right, so let me go through this with you all again. Okay, because a couple of you were a little confused about this worksheet. All right, this picture over here, let me make my screen bigger. So to make sure, whoops, why are you not looking at what I'm looking at? Close. You should be looking at, that's weird. I just had the class up and, so were you even looking at the announcement that I had? Let me try this again. I just made my screen bigger and just noticed that you did not have that up there. All right, so let's try this again. All right, for the benefit of those who just popped in, I clicked on this and I said, make sure that your Chromebooks are functional this week. You need to check on it. You need to let Miss Card know if there's anything at all wrong with your Chromebook, because you have to use these Chromebooks to do the milestone exams, and they have to be in good condition. Otherwise, you won't be allowed to take the test that day, and you'll have to come back on a makeup day. So you don't want that to happen to you. So you'll make sure that your Chromebook is functional. And this scribe here will take you through a Chromebook check. Okay, things that you can check on to make sure everything is working the way it needs to work, so that when you show up for the exam, you're not stuck. All right, karyotyping. Now it's showing. Okay, I can get this out of my way. That would help. Is my volume up? Let's see. Is my volume is up as high as it can go. Chris, so check your volume, Chris, to make sure your volume is up. Move the mic closer to my mouth, and now I'll try to talk a little louder. Is that any better? All right. Thank you. Thank you. All right. So for the first question, this box right here, let me go back just a little bit for a sec. All right. Okay. So this on this page in Canvas gives you a picture of what, and these are all straightened out. Okay, so chromosomes aren't straight little popsicle sticks like this. They have curves and curls and things like that. So these, they straighten them out just to create a picture that you might understand. What you have to understand about this is you have two sets of chromosomes, one from each parent. And these are all different sizes because they all contain different informations about you. Okay. And there are 23 pairs, except 1 through 22 are what we call autosomes. Those are the characteristics that describe what's going on in most of your body. The last pair, the 23rd pair, are the sex chromosomes, and they have all the information about maleness and femaleness. Okay. And they can either be, so this one says either or, they can either be an X and a Y or two X chromosomes. But again, you get one from mom, one from dad. Dad is usually the determining factor because he's the one that's going to give an X or a Y. So if he gives an X and mom can only give an X, then we have a girl. If he gives a Y and mom gives an X, we have a boy. Okay. All right. So on this side of the picture, it says what they look like normally. This one is called trisomy 21 because if we look down at chromosome number 21 down here, there's 3 of them. Okay, so this is what you had to do with that worksheet. You had to look at each picture and go through each set of chromosomes to determine were there any defects and where the defect was. All right, so on this first one, and if you haven't done your worksheet, you better be listening now because I'm giving answers. All right, so you're going to look at the karyotype to the right, and you're going to circle the autosomes. The easiest things, most of you just circled each individual. What you could have done was just gone from 21 to 22 and circled all of that, because all of those are autosomes. Okay. And folks, some of y'all coming in real late these days, and then you come in just for attendance sakes, and then you log out and you're not doing your work. And I'm taking note of that also. But you're late. All right. So again, you could have did a big circle around 1 through 22. The 23rd one here would be where you would have put a box. And when I grade the worksheet, I'm looking for the box and the circle for this one. And it says, is this person male or female? This one it tells you right here, there's an X and a Y. And then you see one tall and one short. This is a male. Okay. How many chromosomes does a normal person have? And this is a fact, 46 chromosomes. Now, there are 23 pairs, but it says total chromosomes. So you got to read these questions carefully. And this is a test strategy as well. While you're taking these tests, don't be in such a hurry to read the question and then get it over with. Read each statement carefully, because this could have asked you how many pairs of chromosomes, but it didn't. It asked you how many total chromosomes. So there's 46 chromosomes all together. And it said put a star next to a homologous chromosome. You could have put the star next to any of them because all of these are homologous. Homologous means they are the same. So number 1, these are 2 homologous chromosomes. Okay. All right, next one. It said look at the karyotypes. Now we're looking at the second one underneath. All right. Do you see any chromosomal abnormalities? And if you looked real hard over here, you had an abnormality where you had X on the 23rd pair. There's 3 of them. There's only supposed to be 2. And so you should have said yes, and you should have circled that one. And then using the key below, identify what's wrong with this person. So if you lose this box down here, if you have 3 X's, the disease was triple X disease. And again, if you don't have your worksheet handy and you're not filling these answers in, you should. In addition to, if you've already done it and I've graded it, you need to be looking at this as well, because some of you didn't get a 100. And this is prep for the test. They will give you a picture of a karyotype. You need to be able to recognize what you're looking at. They will probably give you a key, so you won't have to memorize this. They usually, things like this, they give you a key for that. But you still have to be able to find it, know what the abnormality is, and then look down on the chart to see what it is. All right, next one. It says, look at the karyotype to the left. So that's this set. Is it male or female? This one down here. So we get the 23rd one is always whether it's male or female. So you didn't have to go through looking at all of them. You look at the last one. The 23rd pair is always, when they set these maps up, they always set it up so that the 23rd pair is the sex chromosome. So this is an X and a Y, so this is a male. Which chromosome is the longest and has the most genes? Now, this is kind of what they look like. The one on Canvas had everything all straight and pretty, but chromosomes don't look straight and pretty. They look like this. So the one that's the longest, and it's kind of a tie between 1 and 2. So I would have accepted either one of these answers. And when they're the longest, they also have the most genes on them. So each one of those little bars on the chromosome represents a gene. All right. So so it says 1 or 20, 21. Since you had a choice, 1 was the answer. Which chromosome is the shortest? 2 or 22? Since 2 is long, 22 is your answer. They're real short. All right. In a normal karyotype, how many autosomes are there? Autosomal ones, again, are the regular body cells, not the last two. So you could have told me the answer could have been 22 pairs or 42 total chromosomes. So that either the answer here could have varied depending on whether you were giving me, because it said normal karyotype. It didn't ask you how many pairs or total. So I would have accepted both answers. In a normal karyotype, how many sex chromosomes are there? 2. Okay, you have an X and a Y. One type of chromosomal mutation is trisomy 21. What is another name for it? So we come down here to the chart. Trisomy 21 is also called Down syndrome. Okay, this one was normal. There were actually, this one was not normal because on 18, there was an abnormality, but they didn't ask you about that. But 18 has an abnormality. And down here, if you look down here, that would have been Edwards syndrome. There's 3 of them. Trisomy on the 18th chromosome is Edwards syndrome. All righty. So we're still going to need that box up there. So then you had to look at each one of these pictures. It said identify the disorder and decide whether it was male or female. So you had to circle that and look at it. So in picture A, we look at all the chromosomes. And do you see anything wrong with this?
There is a single X and there's no Y.
There's a single X and there's no Y. All right. So we come back up here to our chart and we got a single X, and that's Turner syndrome. Okay. Just an X, no Y. This is a male. Okay. Identify. So B, we want to look. And what do we have here?
A single X and 2 Ys.
Single X and 2 Ys. Single X, 2 Ys. Jacobs syndrome. Also a male. Okay. All right. Next picture, C. So we look through. What do you see?
Two X's, no Y.
Okay, that just so if you have 2 X's and know why this is definitely a girl. So this is a female, but that's not where our problem is. Our problem is over here with number 13. Okay, so number 13, we come back up to the chart. There's 3 of them. So yes, thank you, Jocelyn. It's trisomy 13 and it's Patau syndrome. Okay, so everybody see that number 13 has 3 of them. You're not supposed to have 3, you're supposed to have 2. So over here on the female one, just two X's is good. Not having a Y means that you're a girl and not a boy. All right. Why is this person coming to this class? I'm not going to let them in. They'll figure it out. Okay. Where are we? We're on D. All right. So we've got, if we look at the last one, there's 2 X's. So this is a girl. But our problem right now is right over here.
There's 3 and 21.
3 and 21. So we come back up here, 20, trisomy 21, Down syndrome. All right, and the last one. So yeah, we have two over here and 1 over here. That's not good. So we have 2 X's and 1 Y. 2 X's, 1 Y, that's Klinefelter syndrome. Okay. All right, so I have just gone through the whole worksheet. If you haven't turned yours in, you just had all the answers, no excuse. All right, what we're going to do now is, where did I put it? I didn't put it in the module. I didn't exactly know where to put it. Okay, it's on your student view. It's not on my page. All right, so on your page, you should have something that says Biology Milestone Test Practice. And Desiana, you logged out so quickly. Mr. Phillips had opened the test back up for you, but for those of you who didn't finish the benchmark, we're going to worry about that on Wednesday. So I'll round up all of you who need to finish it. And then, like I said, after the break, we'll assign you practice things to work on depending on your area of weakness. All right, so if you click on this, you'll come here. Now you can do this anytime you want to. We're going to do this one over here, practice tests, guides, and samplers. That's what you want to click on. So you should do this, do this, do this. Everybody should be doing this. So do I need to go back? Everybody should be doing. Jail, I don't think so, but we'll worry about that on Wednesday. So try to be present on Wednesday, please, because Mr. Phillips has to open the test. They are locked and he has to open them. So I would much rather you focus on this today, and we'll worry about the benchmark on Wednesday for those of you who need to finish it. All right, so everybody do this, please. So let me come out of it and let me start back again. All right, so on your, oh gosh, why does it do that? All right, so on your home page, everybody please follow. You should have something that says Biology Milestone Test. Is there anybody who does not have that? All right, click on it. Mine's slow now. All right, so they have offered this to you. So this is a link, like I said, that you can use this to practice anytime. And you want to hit the button that says practice tests, guides, and samplers. Click on that again. You are taking the end of course. You want a practice test, and you want standard online tools. And we want biology. Okay. Now, This is exactly what the test will look like. When you log in, it will have your name and you'll have an ID number and all of that. These are practice tests, so it has general information. But the first thing that they'll be asking you to do is check to make sure that the information they gave you matches the information on the test. Otherwise, it won't report your scores correctly. And then it goes through all this information about how the test will look, things that you should be looking for, and then it'll give you test directions. It will tell you to read, look for pointers, and then tell you how to hit the next button each time. So you should read all of that. Well, actually, the test examiner usually goes through this part with you. I'm sure all of you have taken milestone tests before, so you know that the examiner usually reads this part to you. This part too. And I'm skipping through it because this is practice for us. So they will go through all of it, but this is what the test will look like the day that you set up. So then we're going to just begin the test. Okay, this is a practice test. So this is what it's going to look like. So we're going to go through a couple of these questions and then I'm going to stop and and actually, I'm going to let you look at this for a minute and I'm going to stop sharing. I'm going to come right back to it as soon as I do attendance. I don't want to get so caught up that I forget to do attendance. So you look at that and I'll be right back so I can log in all these late people. And again, folks, the focus mode is working so you can come on camera. I found where they hid it. They changed the whole format of the Zoom page. I had to figure out how to get the participant and chat box not to be blacked out. Figured that out. But all right, wait a minute. Whoops, I'm doing that all kind of wrong. Okay. Why do I have so many people on my list? Somebody is missing. All right, everybody should be practicing. I'm looking at people and this screen says idle. Everybody should be on the practice site. I don't know why you're not there. Now, this is to help you. This isn't for me. I don't have to take the test. This is to give you advanced look at what it's supposed to look like. All right, I figured out my missing person. And again, folks, we're going to be practicing and doing answers. And those, like about 5 of y'all were late today. So y'all just try to be on time from here on out. Because the only reason I went backwards today was because my screen was not where it should have been. All right, so when I go through these questions, I'm not just looking for an answer. I'm not just going to run through and give, I'm going to explain because this is a sample test. This is not what the question you might get may not look like this. Okay, but it'll have similar information. So we're looking at what kind of topics are going to be on the test when we do this. All right, let me get my screen big again. Okay. All right, so this is a genetics question. This is a meiosis, mitosis question. And this is one of those questions that you all had a lot of trouble with, looking at what the chromosomes were doing, where they were located, and what this information is actually telling you. Okay, so we look at this. The first thing that, even though according to this, these are popsicle sticks and Q-tips that they are using to represent a cell. So if you were in the classroom, we might have did this activity as well, where on a plate, paper plate that has a rim to it, the plate represents the whole cell, and in the middle, we have, in the number one, you have a circle around our sticks, and this would represent two homologous chromosomes, because they're two of each, like you have the popsicle stick represents one kind, and the Q-tips represent another kind. Okay, so they have different information on them, but there's two of them, one from mom, one from dad. All right, second picture. Can somebody tell me what's the difference between picture 1 and 2?
One has a circle around the chromosomes, and two, well, first of all, in one, the chromosomes are together in a circle in the nucleus,
Mm-hmm.
and in two, the nucleus disappears and the two chromosomes
Thank,
separate.
thank you, thank you, thank you. That's exactly the way I wanted you to say it. I want you to call that circle a nucleus, because that's what it is. All right, so he said that the nucleus disappeared in picture 2, and they have started to move away from each other. All right, in picture 3, they're kind of lined up in the riddle. So you got to remember your, what do you call the thing? PMAT. Okay, prophase, metaphase, anaphase, telophase. All right, so that's what's going on here. And then we've pulled the homologous pairs have been pulled apart, so there's one of each on each side. By the time we get to 5, the nucleus is coming back, and by the time we get to 6 and 7, we have created 2 new cells that look like number 1. Having said all of that, the question says, which statement correctly uses the model to explain how mitosis maintains genetic continuity? That means that we should end up with what we started with. Okay, we want to end up with the same number of genes that in each cell that we started with. We started with 2, one of each kind, and over here we have 2 new cells, one of each kind in each cell. All right, chromosomes in cell 1 are the same as chromosomes in 6 and 7, and I'm going to tell you that that's the right answer. Because if you look at this, I think this is what I just said. This was what we started with. And when we finish, we have 2 cells, because mitosis is about creating 2 new cells, but with the same information. So these are identical. These 6 and 7 are identical to number 1. Okay? All right. So all the rest of these are wrong. And I'm skipping through it because I've already read the question. During a test, you need to read the rest to make sure that you are examining each of these and you understand why the answers are wrong. Okay? Uh, they use the term crossing over. You need to know when crossing over happens, during which phase of mitosis crossing over happens. What's the other word? Independent assortment. You need to know what independent assortment is as well. And what does that have to do with these separating? All right, so then you're going to push your button to go to the next question. All right, this question is about our macromolecules. Way back at the beginning of the year in August, when we started school, we talked about macromolecules. All right, this macromolecule is made of chains or rings of carbon atoms bonded together with hydrogen and oxygen. That's our CHO. All right. So if we look back at our handout that we had function, we had a whole little graphic organizer that had function of each of the macromolecules. This describes a carbohydrate, and carbohydrates function is short-term energy. You get quick energy um, from carbohydrates. We were saying that like a lot of times football teams they have a nice spaghetti dinner before a football game because those carbs will give you some energy to get you through the game. All right, next macromolecule is made up of 1 glycerol and 3 fatty acids. So that describes, so if you looked at your chart again, and that would describe that they are made up of long chains of just carbon and hydrogen, very little oxygen, if any. But the fact of the matter is its structure is 1 glycerol, 3 fatty acids. That's a fat molecule or a lipid molecule. And you get more energy from a fat molecule than you do from a carbohydrate. So both of them will give you energy. But if you want long-term energy, you want to eat fats, not carbohydrates. That'll give you quick energy and then you crash. All right, catalyzing biochemicals are enzymes. And storing and transmitting genetic information, those would be chromosomes. Anytime you see genetic information, those are chromosomes. Okay? All righty, that's that question. Any questions about that? Stop me if I'm confusing you. This, like I said, this is review. So if there's something about this that you don't understand, please stop me. All right, this one is one of those questions that we have not touched on evolution and ecology yet, but we need to know the term invasive species. So when we come back from break, we will talk about ecology and invasive species. And invasive species are species that are not normal to an area, but they set up house, and then they usually kind of take over and out out compete with the local species, so they cause damage. Okay, one of the characteristics that we have here in Georgia is the kudzu. When does what end? I don't know what SB. Unmute and ask your question. Oh, spring break. Spring break starts Saturday, and you come back. You have 1 week off for spring break, so you will be back on April the 6th. All righty. So again, when we come back, we'll spend a lot of time talking about ecology, taxonomy, and evolution. A lot of time, we're gonna do snapshots of those topics because we haven't spent, and then we'll come back after the test and do a little more with those afterwards. All right, so again, invasive species are those kinds of organisms that move into an area where they're not native to that area, but they outcompete for food and shelter and all that kind of stuff. So the things that are local kind of either die or have to move away. And I was saying in our area in Georgia, we have the kudzu plant. Kudzu is that green vine that grows on top of the buildings and the poles and trees, and it has just taken over everything. And it mostly lives on other trees and just saps the life out of it. All right, so the question says, in this case, some insects bore holes into live trees and then get transported when the tree is cut down for use for numerous projects. Wooden crates used for packing materials may be infested with insects or eggs that have been transported over long distances. What is one safe, practical way that people can act responsibly to prevent spreading potential harmful invasive insects? This is where you're going to have to read each one of these carefully. All right, store firewood from one winter until the next winter in a shaded grassy area to starve the insects and stop propagation. Well, we're not exactly dealing with firewood, so that's not helpful. Spray wooden products with pesticide before bringing them into your home or buildings to kill insects before they enter the structure. And you could do that. Chop down trees suspected of being infected and use them. Wood chopper would make them safer for mulching landscapes. Buy local firewood near the campsite instead of transporting raw materials to another area, and then leave any extra for the next camper. This, I feel, is the best answer because you're not bringing something from one place to another. You're using what is there locally. You're only using what you need. And then anything that's left over, you leave for the next person. You're not trying to say, I'm going to take all the extras home with me. Do not take the extras home with you. Leave them right there on the campsite. So I would have picked this one. Okay, so that way, because it says to prevent harmful spreading of infectious insects. So if the wood had anything in it, you're going to leave it right where it is. You're not going to bring it home with you. Okay. Anybody disagree? Okay. All right. A model. Now we have done this. A model process of tRNA. Remember that with protein synthesis, we had messenger RNA, ribosomal RNA, and transfer RNA. So each one of them had a different job. So which one best explains the role of tRNA? So again, you need to know that T stands for transfer. And if you can remember that T stands for transfer, then you can remember that its job is to transfer things from one place to another. In this case, we are transferring amino acids to the ribosome. So that's kind of what the picture is showing you. This thing has the little circle at the top represents our amino acid. It's attached to our transfer RNA, and it is bringing it into the ribosome where it will connect. And once this one fits in that little space there, the two amino acids will connect and the transfer RNA will go on its way and go get another one. Okay, so A is the correct answer for this one, but I'll go through the rest. All right, tRNA recognizes the stop codon of a developing peptide so that no new amino acids are added. It will do that. But it's also the primary function of this is to bring, so it will recognize the code and the code will say stop and it'll tell it don't get any more amino acids. And so it was, like this one was start. So this one knows that this is the beginning, and if it was stop, there would be nothing here because it would not bring an amino acid to the site. The way this picture is showing you, this one is bringing the amino acid to the ribosome. So this will confuse you because that's not a wrong statement totally. But again, its job as far as this picture is concerned is to deliver amino acids to the ribosome so it can be added to the developing line. tRNA signals the release of the peptide. No, it does not. tRNA scans the developing peptide to make sure the sequence of amino acid matches. It does not do that. So these last two are definitely wrong. B was set there to confuse you, but A is the correct answer. Okay, because that's what the picture is representing. It is delivering an amino acid. And again, amino acids are usually represented by little circles. This whole blob right here is the ribosome. This line represents the chain, the peptide chain that it's trying to form, or the conveyor belt as the amino acids are being formed. All right, we didn't spend a lot of time talking about bacteria either, and antibiotics, but you can answer this question by looking at the picture. All right, so we had one and then it multiplied. Okay. And then somebody gave it an antibiotic and then it looked like it died. And then all of a sudden, we got twice as many over here. Okay. So that's what the picture is showing you. So it says, which sentence best explains how the indicated step in the model allows bacteria to develop resistance? Okay, genetic mutations that promote resistance occurs. The bacteria are infected by viruses that concur with the resistance. Alleles for antibiotic resistance become dominant over recessive alleles. A portion of the genetic material is re-replicated, causing resistance. This A and D are interesting answers because genetic mutation, this is not necessarily a mutation, but a part where the replication process, like you said, the portion of the genetic material is re-replicated. So some of it, and then viruses and bacteria operate a little bit different. And so they cause the resistance. So if portion of it, because when the ones with the X's seem to be the ones that didn't die. So some of these cells did die. These are not mutated. These are ones that didn't seem to fall down at all. And then the ones with the X's seem to be the ones that got replicated. So, okay, so they were replicated and that caused the resistance, and then they were able to make some more bacteria. Okay, so I would pick D as an answer for this. Anybody disagree? Okay, so you got to study the picture. You had cells with circles and some with X's. And then over here, all the circles seem to be taken down by the antibiotic, but the X's seem to be alive and well. So those survived and they were able to replicate, and they caused and they developed a resistance to whatever it is you gave the guys with the circles. So that means you got to come up with something new. Oh, look, a Punnett square. All right, it shows inheritance of a flower color for a snapdragon. And here again, they tell you what the key is. They're not just going to give you letters and not tell you what it represents. So the capital R's are red flowers. That tells you that red is dominant. See, they didn't say that. They just show you capital R. But you need to know capital R means that red flowers are dominant. Lowercase r's means white flowers are recessive. And if you have a capital R and a lowercase r, you get pink flowers. So this one is our codominance. Remember, you can have codominance or blended dominance. All right. Let's see. The alleles for the flowers show, and that's what I just said, codominance. Both of them are dominant, so that we're getting pink, and we're not having incomplete. There's a whole different variation of, actually, look that one up, because let me look that one up. I don't want to tell you wrong. Both alleles are completely expressed in heterozygous individuals because they're not the same. Well, now, wait a minute, let's look up what is codominance. And you won't be able to do this on the test. But okay, so it is incomplete. Unlike so incomplete where red and white makes pink. All right, so let's go back to that question. That's why I was quite sure and I didn't want to tell you wrong. So this is incomplete. Codominance was our calico cat, where we had that totally different situation. Okay, so this is incomplete because both alleles are completely expressed. Partly expressed in heterozygous individuals. There we go. Right? Okay. All right. So again, vocabulary. If you do nothing else over the break, study your vocabulary. You can practice these tests, but you're not going to have these exact same questions. It's nice to do practice tests, but sometimes practice tests lead, because then you'll say the questions you had as practice were not on the test, and they're not going to be. Okay, these questions are designed to give you an idea of what the test questions will look like. And so you need to study the topics and the vocabulary, but not study the questions. Okay, here's another ecology question. Ruby-throated hummingbirds are found throughout North America. They are omnivores. So this has to do with our food web and food pyramids and food chains. So again, after the break, we will spend a lot of time talking about those because those are important. There's a lot going on with those diagrams. Omnivores mean they eat everything. So again, vocabulary, no vocabulary means there's omnivores, carnivores eat meat, herbivores eat only plants, and let's see the other one. Omnivores, carnivores, and okay, that's the three. And then you have your the detrivores, which are the decomposing. They feed off of decomposing material. So I knew there was another one. All right, omnivores feeding on nectar, pollen, tree sap, insects, and spiders. Okay, so that's plants, animals, all in one thing. They get their water from the nectar. Most nests are in forested areas, citrus groves, marshes, or shrubland. The hummingbirds breed in the spring and the summer, and the females raise 1 to 2 broods per season. So, a brood is they could lay eggs in here and then they grow and they lay some more and they grow again. All right. Most migrate to Mexico, Central America in the winter. They fly nonstop for 20 hours across the Mexican Gulf. However, increasing number of hummingbirds have begun to winter north of the Gulf. Researchers have documented that hummingbirds' winter range has expanded northward about 30 kilometers in the recent years. Which task is most important in the ability of hummingbirds to survive their first winter? They need to find suitable nesting areas and sites where the most important task of the hummingbird for the hummingbirds because they must be able to produce offspring. Finding sufficient supply of flowering plants, flowering trees, insects, is the most important task because they need food to survive. Finding bodies of water, important to the task because they need supplements of the nectar if insufficient to provide all the calories they need. Finding mates to allow them to lay eggs in fertile areas. And I'm not going to read the rest of that. All right. They will, if they don't have the strength to fly, so they need food. Okay, so yes, they need to find nesting sites. That's important because they need to lay eggs. But first of all, they need to find flowering plants. The winter is going to determine whether or not they find flowering plants. okay? Because they need food to survive. If they don't have enough energy to survive, they're not going to be making nests and laying eggs. So they first need to find flowering plants. So what is happening with our seasonal change now is that it's being warmer and warmer, longer periods of time. So they're finding, like they said, uh, northward, it's getting warmer a little earlier so they can survive in areas that are not going to be as cold in the winter. But if they're not producing flowers, then they're not going to be able to find food because that's where they get their food from and the nectar. So even this one over here with the water, they need water, but they mostly try to get that's to supplement the nectar. So they need to find flowering plants, trees, and insects for food. Otherwise, again, they won't have energy to do anything else. Reproduce, fly, make nests, all of that requires energy. You need food for that. Okay, so they got to find this first. And if the winter is that time of year where some trees don't flower, insects are not around, so if they're not going to find any of those things, then they won't stay there. Okay, so this is one of those questions, even though it's an ecology question, you could have figured that one out just by the fact that they need food to survive. And some questions are like that. You can figure them out common sense. We're going to talk about this too. So this is also something that we have not talked about, but when we do taxonomy and ecology, we're going to look at this cladogram. And this line here is a general connection, but they're going to tell you that the lines that are closest together say that they are more closely related organisms. So in classification, invertebrate means anything that doesn't have a backbone. We are vertebrates, we have an internal skeleton. Invertebrate means that they don't have internal skeletons, but they might have shells on the outside, okay? But they don't have an internal skeleton. So if you look at this, even though all of these are invertebrates, the worm is nowhere close to being related to this dragonfly. So the ones that are closer together represents some type of similar ancestry. Okay, so they're organized by similarities and differences shows that the relationship between some invertebrates based on their body structure. And then you can create these in different ways. This one is based on body structure. Use the drop-down menu to argue the development of six-legged invertebrates based on the body structure shown. Okay, six legs can be placed. All right, so we're looking at these letters. How many legs? They got 8 legs over here. They got 6 legs, A and D. So the development of six should be placed with D. On the cladogram because the body structure represents only an organism. Right, so everybody after that point has 6 legs at organism after that point. Okay, everybody after this point. Now that's a good question. We're going to say E because these two are kind of closely related, but everything after this has 6 legs. So you could argue that it was D because but D is also in this area. So everything after this, because everything represented after that point. So everything after that point, after E, has 6 legs. Okay. And hopefully your picture will be a little bit bigger than this so you can see the difference between Mr. Ant and Mr. Cricket. All right, I'm going to stop right there. And we'll do another one on Wednesday. Actually, I'm going to let you do it on Wednesday because I do have some folks that need to do the benchmark, and then we'll do another one together on Thursday. Okay, so on Wednesday, I'm going to let you go through it by yourself. And we'll get to the folks who need to do the benchmark. And then Thursday, we'll continue doing it together. Okay? Questions, questions, questions? All right, so you have the link for this. It's on your homepage. I would copy it and move it somewhere else. Actually, I stopped sharing too soon. I want to go back because you can. Okay, I'm going to share again because you can do the test and you can do. You can do the study guide as well. So if you get study guides, that might be more helpful because it will give you a general overview. And study guides tend to be updated, so it will have the topics most likely on this year's. Okay, so if you do it like this, it explains the answers for you. Okay, then you see this one is a totally different question than the ones we were looking at. So it gives you a variety of things to look at. So you could do a sample test or you can do the study guide. So Thursday, we'll do the study guide. All right. All right, if you have no questions for me, you are welcome to log out. And I see somebody already did. All right, questions, questions, questions. So I hope you all were paying attention. Some of y'all were still off camera. I don't know what y'all was doing. But again, this is for you. And you got to pass this test. You got to pass this test. So it's to your benefit to study. Study, 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 study. All right. Have a good afternoon. See you on Wednesday.